Claudia, there was a question about your logo, which is quite terrific. Do you know who designed it? Was it done?
Yeah, it's a student who did a design degree first.
Oh.
She worked for a friend, and so we approached her. Don't know if giving up a design career is better than having a biology career. I don't know, but she made a very nice logo for us. Sorry, I didn't hear that question. Maybe it was in the chat. I didn't.
It's in the chat, yeah.
Thank you.
Okay, so still 2 minutes to go. So maybe all the people that are non-speakers, can you mute yourself in case that's not the case yet, so that we only hear the speaker?
And as a reminder, if you have questions, you can raise your hand. There's a raise hand button at the bottom of your screen. And you should also, if it's not on the bottom bar, you can click more, and there's a Q&A feature. So you can also type out questions there that can be answered during the talk. And you can also, of course, use the chat, both for questions, or if you have any tech issues, you can type those to everyone or to me, and we'll try and resolve them throughout the presentation.
Okay, 1 minute to go. We're almost there. Okay, so I will start with welcome you all. So thank you all for being here. So it's a great honor to share this STN network together with Franjo, and it's also a great pleasure that you all join and come in here to our STN networks. So I give the word first to Claudia Fricke on the evolutionary ecology of thermal fertility limits. There will be 2 talks on this network. So go ahead, Claudia.
Thank you very much. Nice to see you as squares. So we are a network and we will have 2 slots. So this one will be more about how we approach forming this network, and then the second talk will be more about the science given by Liam. And so what first of all, who we are. So we're not just a pretty logo, we are also some pretty faces. So we have a leadership of 5 people, and we come from different countries. Well, with Britain bias, but there's also Rhonda from Stockholm, and I sit in Germany. And so we came up with this idea and proposed it to ESEB. And the idea was based on some data that Rhonda, Tom, and Amanda with their team collected, and this has been published for a few years now. And they did this about 40 plus Drosophila, where they measured where the lethal limit is, and these are the black circles that show the lethal limit for each species, but then they also looked at where the fertility limits for males is, and those are the pink circles. And the idea that males are sterile when they're exposed to high temperature has been shown in Drosophila before, but here the idea of having limits comparable to the critical thermal limits was explored. And it showed that these thermal fertility limits, as they were called now, are really quite important in determining species distributions. They're quite good predictors. And as most of the climate scenario models are looking at CTLs or something similar, like performance curves, we thought this might be something that is of importance but is potentially overlooked. And so this was sort of the basic idea that started us off with forming this STN, this network. And so what we set out to do was, first of all, to provide a forum for researchers who are interested in these ideas about thermal ecology and the evolutionary aspects of the challenges that animals face at the moment. So we are only focusing on animals, as we are animal biologists, so we can only encourage plant biologists. We have a few on board, but we haven't managed to merge the two successfully. And so what we thought, as this is an emerging field, we hope is to, first of all, advocate for best practice methodology. I will talk about that one in a little while. And so we want to inspire data collection to set this on a broader basis, particularly a broader taxon base. And we also think that theory development can also be of importance here, and especially exploring links with modeling approaches for future scenarios is something that we would like to explore much deeper. We are currently a little bit over 70 members, and for Europe, the little axis can tell you where we are. So I think we have a really good spread across Europe. We are missing a bit the eastern side of Europe. We also have some international, well, non-European international members in our assortment. So we are open also for non-EU members. And yeah, we are basically steadily growing and having people ask us. And yeah, so if you're interested, just write one of us an email. So we got awarded the Special Topic Network in August 2019, and we were planning our first inaugural meeting in March. And while we were writing the invitations, we started to worry about what's going on and should we travel, should we invite people? And it very quickly then became clear that this is not an option any longer. So we had to think about other measures. So what we did is we actively invited people we thought are interested in this topic. And then we held, because Zoom at the time was new and we didn't know how many people it can handle. So we had 3 different meetings where we had different subsets of people that we invited. We invited for a discussion, we presented our ideas, and this is how our network got started. A fraction of those were actually interested. And it was actually just about 2 years later that we all managed at least partly to meet in person. So we had our first kickoff meeting with a 2-year delay at ESEB when it was in Prague. So what we did to communicate have being spread across all these different countries and needs that we communicate quite well with each other. So first of all, we established also a web page to communicate with others outside of our network as well, and to make this more broadly available to also interested people. While we started out with WordPress.org, .com. We now recently, earlier this year, moved to .org because we noticed that it allows us more freedom. We needed a local host here. So just as a little tip, if you start a web page, maybe consider .org. We mostly communicate via email, so we build an email list that is, as said, steadily growing, and this came through the university IT unit that they had mailmen available that allowed us to maintain this mailing list at not too much yeah, it doesn't take too much time to do that actually, but allows us all to send emails to each other. We had Slack in the beginning as a more sort of tool that can be utilized more easily, and we can exchange messages. But as Slack changed its policy and we lost all of our information after 19 days and weren't willing to pay, we now moved on to Discord. And after some first little hiccups, it's now working really, really well for us, and it supports our work quite efficiently. So also that is only something that can be recommended for other people who have to run a network. So what are we actually doing? We have monthly seminars, and as we only had one in-person meeting, mostly to discuss things, and as we are so spread out, we thought we also need to foster exchange between our community members. So we often include internal STN members and also give PhD students a chance to then present their work so that we increase communication between our members. But we have also actively invited external speakers, on interesting topics. We have annual meetings. If they're not in person, then they're online, so where we exchange and discuss what's going on. We held a few workshops also. More on the specifics will come when I come to what we actually did. And so what we did during the first 2 years as active output is that we worked on this unified protocol that we wanted to provide as a starting point to ideally have comparable data collected across taxa. And we also embarked on a meta-analysis effort. And so our unified protocol actually just got published earlier this year in the Journal of Evolutionary Biology. It took us a surprisingly longer time to assemble this than we thought. But it ended up that we actually don't just have one protocol, but after a lot of discussion and taking on board all the different experiences that people have with their diverse species they work on, led to actually having 3 different protocols in there. So we have one we call the gold standard one that actually is to estimate thermal fertility limits by itself. But then for species that cannot be raised in high numbers because that protocol needs a lot of test animals, we also have a thermal sensitivity of fertility limit. And then if you work with a species you cannot bring into the lab or cannot in the lab, we also thought it's worthwhile to collect data out in the field, and we called that the natural history of thermal sensitivity of fertility protocol. A bit of a lengthy name now that I have to say it. So yeah, we hope that encourages people to collect this data, but that should only be the first step, because what we think is necessary is also to build up a database. So this is our following on next step from having this method paper, as we call it. And there we were lucky that within the network we actually have people with experience who have built databases before, because we in the leadership team lack experience. And we were even more lucky because there is an initiative of some of our members in Amsterdam who built up a really professional database, and they are allowing us to sort of use their infrastructure for the future. So that is something we're building up, so that hopefully then we can also start collecting this data and have or add our data to this database that they are collecting. Our second major initiative was this meta-analysis effort that we that I mentioned earlier, and this is just representation of where we started from. So we screened the literature that's out there, and these are the species that we found. And what we did is we wanted to see what experimental data between fecundity, fertility, and temperature effects on these reproductive traits is out there. And we started off with close to 17,000 papers, and ended up screening over 40,000 papers. And this was only possible because the community was really active in helping us. So I guess we were around 30 to 40 people who actively did the screening and then mapped all the data of those articles. So close to 1,600 articles survived our screening process, and so we could map the data from this 1,100 species. Also, that got published earlier this year, and this is our basis for our meta-analysis efforts. So one big meta-analysis, we just submitted the manuscript I think a couple of weeks ago, and because this really was a big effort, we had one workshop that we called the Data Extraction Extravaganza that happened more than a year ago, where everybody took out a week to actually help with this massive efforts. But having this map, we also saw that there is more questions we can ask than we initially thought. So at the moment, we have 13 different meta-analysis efforts going on, and inspired the community to start posting and addressing new ideas. But having done this, we also encountered, I think I'm running out of time. So we actually encountered a lot of problems. And the most was how do we manage expectations and overlaps between a lot of running projects. So come and ask us about this. Authorship policy is the keyword here, and guidelines. We could only meet these challenges because we were also lucky to have a lot of experienced people on board who could help us with this effort. That was more massive than I naively thought before. And we have future projects going on. I'm really sorry I managed my time so badly. And also there we are lucky to have experts on board, because we now want to do a wild monitoring that should go on a yearly basis and across Europe. Thank you very much for bearing with me, and yeah, I take questions in the Q&A session.
Thank you very much, Claudia. You actually managed very well because you're just on time, so
Okay.
not too much time overflow. So thank you very much for that. So I think in the meantime, maybe we can switch the slides, and Liam, can you share?
I just leave.
Yeah, let me reply.
If you have already questions, don't hesitate to put them in the chat or in the Q&A option. That's also possible. So Liam, go ahead.
Okay, can everyone see my screen?
Yeah.
And can you see my mouse as well?
Yes.
Okay, great. Okay, I will start. So thank you, Claudia, for introducing the project, and thanks to the organizers for giving us 2 slots. So what I want to do is quickly talk about the main meta-analysis project that we've just finished. And as Claudia said, there are many others going on, but this is the one that has the furthest progress. And so before I begin, again, I want to thank lots of people. So almost all of the names here were people who helped extracting data from these papers, and they did that voluntarily just through the network, which is amazing. Facilitated by these weeks where we all got together and we had training and we extracted data. But then a lot of it was also led by my postdoc, Faye, who can't be here this week, and she was funded by the Natural Environment Research Council. So she really helped to collate all the data, run the analyses, write the paper. Okay, so to the science. So one of the ways that we can try and predict how climate change is going to affect natural populations is to look at thermal limits in the lab. So we take animals into the lab and we look at the temperature at which they die, and we would call that a lethal limit. The temperature at which they fall over and stop moving, and we call that a critical limit. And more now, we're realizing that we also need to look at fertility. So the point at which an animal becomes infertile or completely sterile, and we call those fertility limits. And work as part of the network 3 or 4 years ago now, took a systematic approach looking at lots of different Drosophila species, and they recorded the lethal limits, which are the black dot, and the fertility limits, which are the pink dots, across 40 species. What they found was that often the fertility limits are significantly lower than the lethal limit, right? So there are, and this is male permanent sterility. So these are males that are becoming permanently sterile, in some cases several degrees below the point at which they would die. And when you map that to species distributions in the wild, you see that the fertility limits are a significantly better fit. So it seems like in terms of fitness, it's this lower limit that's more important, because of the lower limit makes the male sterile and the population crashes. So really cool study, really high profile study. And so the question is, how general is this effect, right? We only really have this type of systematic comparison for fruit flies. So that was one of the things we wanted to test or answer with the network. And to start doing that, we made this systematic map. So we basically systematically searched the literature for any studies we can find that link temperature to some type of animal reproduction. And we found, as Claudia said, 1,600 studies, over 1,000 species. Lots of species, loads of data there that we can work with. Problem we had was that very few of these studies actually push animals to their limits in terms of reproduction. We have lots of estimates of limits in terms of survival, but not reproduction. So what we're left with actually is a data set with not always very extreme temperatures. So in light of that, we decided to shift focus a little bit, and we decided to look at reproduction from a life history perspective rather than a sterility perspective. So we can think about thermal performance. So most of these, all animals have different traits, and they all have a different thermal performance curve, and they all have a different thermal performance optima. And we can look in terms of reproduction at sterility, the point at which an animal is infertile, but we can also look at reproductive output. And the key trait there is fecundity in terms of either the number of eggs that are produced or the number of offspring. So we can look at fecundity as a life history trait and see how that changes with temperature within the region where they are still fertile. And then if we want to look at survival, we can't look at the number of animals that die within a certain time frame, because a lot of these temperatures are not so extreme. So instead, what we do is we can look at lifespan, right? So the average number of days that an individual survived after exposure to a different test temperature. So we've got a slightly now different question, which is reproduction more sensitive to temperature variation than lifespan is, in the same way that sterility is more sensitive than lethality? And so that's what we wanted to test. And really, we're predicting that reproduction should be more sensitive. We often see actually that the range of temperatures that an animal can reproduce is lower than the temperature through which they can survive. So there are hints that this is probably a general trend, but it hasn't been tested systematically before. So that's our question. So we went back through those systematic map papers, and we found any papers that had also a measure of lifespan or survival in the same species that they were also measuring reproduction. We had 830 candidate studies that we screened, and we ended up extracting data for 300 species. And again, this is fecundity and lifespan in the same species in relation to temperature. And here's the tree of the species, what they look like. They are all invertebrates, and we didn't limit ourselves to invertebrates, but that's just what we ended up finding. I think because these are the species that you can actually do these studies in, especially when you're interested in lifespan changes. So animals are short-lived that you can get in the lab. And in fact, it's 75% insects. And I guess the key thing is that they are all ectotherms, right? So they are all animals that don't maintain their own internal body temperature. So they are expected to be quite sensitive to changes in environmental temperature. and also 90% terrestrial species. So we have some aquatic, but this is mostly terrestrial invertebrates. So we took these studies, and then I'll briefly talk about the analysis. The key thing to note is that we expect these thermal performance curves to be non-linear, and they have a single peak, and then they go down either side of the peak. So we have to use an analysis that takes that into account. We can't analyze the entire thermal performance curve for every species because we don't often have that. We often have species that are tested at very different sporadic temperatures throughout that range. So very few studies give us an entire, between 10 and 40 degrees Celsius temperature range. So we don't have curves per se. What we do have is we have set temperature treatment. So the way we deal with that is we turn our set temperatures into pairwise contrast. So we take the average fecundity or the average lifespan from all of the temperature treatment, and then we do pairwise contrast between them. Each time we're comparing the test temperature to some reference temperature, which from our scheme is essentially a temperature which is somewhere in the nice benign temperature zone in the middle. So we're trying to capture a temperature that is close to optimal performance. So here's an example where we have a study with 5 temperatures. We assign some temperature as the reference. Oh, and typically we do that as whatever the temperature is that's closest to the rearing temperature of the animal. The logic being that that's the temperature they are happy at, and they reproduce that in the lab. So we take some reference temperature in the middle, and then we do contrast. So this 5 treatments would turn into 4 effect sizes, 4 contrasts between reproduction or lifespan. And crucially, the model takes into account the fact that these estimates are not independent anymore. We now have 4 estimates from the same study, and they all share the same essential control treatment. Anyway, that's what we do. As I said, the studies use a range of temperatures, and here's what they look like. So we have 2 types of studies. We have studies that are exposing animals across their entire life, the different temperature variations, and we call these long-term exposure studies. They're doing it for more than 5 days, and this is the vast majority of studies, right? They're leaving the animals in different temperatures for a long time. And if we look at this histogram, we can see that the reference temperature treatments are typically around 25 degrees Celsius, and this seems to be pretty consistent, that all invertebrates seem to be happy around 25 degrees. And then the experimental temperatures either side of that, I just want to show you that, one, they're reasonably symmetric. We have warm and we have cool temperatures, but they're also not very extreme, right? So we have very few studies that are going above 35 degrees or below 10 degrees. For these types of studies with long-term exposure. We also have a small subset of studies that do what we would call a heat shock, where they're exposing the animals to extreme temperatures for less than 5 days. And typically it's less than 24 hours. And note that the frequency here is much lower, but you are then seeing more extreme temperatures over here. And very rarely would you heat shock using a cold temperature. But most of the data looks like this, and all of the conclusions I'm going to show you are robust to if we remove these short-term exposure experiments, because they're slightly different. Okay, so what do we find? So I'm now going to plot experimental temperature on the x-axis against the standardized change in reproduction on the y-axis. So what that means is it's a comparison between the reference temperature and some experimental treatment. And because the reference temperatures are what we think close to the optimal performance, our prediction is that most effect sizes should be negative. So if you go above and below this reference temperature, reproduction should go down. So we're expecting this to be negative. And that's what we see. So we see this unimodal curve with a peak around 22, 23 degrees. And we have a reduction in reproduction above about 28 degrees and below about 18 degrees. And the explanation, we think, is that in cooler temperatures, metabolism is slower, the animals cannot get as much resources, and so the reproduction is reduced. And above 28 degrees or so, metabolism is increased, but then metabolism starts to break essentially because the proteins that do things, metabolic proteins stop working. their efficiency is tuned to some intermediate temperature. So they stop working and functions break down. And so we have this zone in the middle where reproduction is maximized. I'll skip this for time. In contrast, the lifespan thermal performance curve looks very different. So again, above 25, we see this decrease. But below 25 degrees, we actually see lifespan increases. And this model actually suggests that peak lifespan, or that should say lifespan, is at a very low temperature and flattens out. Now, I would expect this to go down again, and I suspect the problem is that we have very few data points in this region, and the model can't detect that drop. But again, we have this, so we have a peak at a much lower point, but we also have significant reduction in lifespan above 25. And the explanation for this increase, again, is related to the rate of metabolism. Rate of metabolism is slow and it's cool. For reproduction, that means that reproductive output goes down. For lifespan, it means that the animals start living longer because they're living much more slowly. And so really, in one sense, lifespan is not the best fitness measure at low temperatures, or the best fitness comparison at low temperatures. Okay, and the key thing is, can we compare the two life history traits from the same species? If we superimpose the two curves, you can see that above 25 degrees, the slopes are very similar, but then below 25, it's very divergent. And we can test this formally with what's called a multivariate meta-analysis model. So we combine both traits into the same model. We limited it to species where reproduction and lifespan were measured in the same individual, actually. So we can actually look at individual differences in reproduction and lifespan at the same temperatures in the same individuals. And when we do that, we split it into two sections, basically with 25 degrees in the middle, because these trends are so divergent. And when we look below 25 degrees, we see that the responses are totally different. So reproduction, again, we see this clear decrease. Lifespan goes up as you go towards zero and then goes back down again. So in this model, we are seeing the curvature that we weren't seeing across the whole temperature range. These responses are super different, and crucially, there's no correlation between the two, right? So at one temperature, one species might have a decrease in reproduction, they could have an increase in lifespan, it could be very different. Then when we look above 25 degrees C, what we see is, first of all, both life history traits are decreasing, and the best fit for this model is just a linear trend. We have pretty strong correlation between the two. So a temperature that reduces your reproduction also tends to reduce your lifespan. But the slope for reproduction is more than double the slope for lifespan. So reproduction is being affected by warming significantly more strongly than lifespan is. So this again recapitulates the fact that reproduction seems to be much more sensitive to warming than survival is.
Liam, sorry to interrupt you, but your time is finished.
Yeah,
so
okay.
Can you wrap up it?
Yeah, I'm done. I'm just going to do a conclusion. So basically, just to say, yeah, no problem. In summary, so we use this meta-analysis to compare long-term temperature effects on reproduction and lifespan in the same species. Reproduction seems to peak at a much warmer temperature than lifespan does, but crucially, reproduction seems to also be more sensitive to warming than lifespan. And so again, studies that focus on lifespan or survival are potentially underestimating the thermal sensitivity of these species and the vulnerability to future warming. And yes, I will end there. Thank you all for listening.
Thank you very much. Thank you for this nice overview. So very nice to see this all together. Amandine, I see your present. Maybe you can try to share already your slides, and in the meantime, we'll check if there's a question for Liam. Is there anyone who has a quick question? No, only a very Ed, you have a very quick
sorry,
question.
I couldn't find the hand button. That's very interesting. Thank you, Liam. So just a quick question. I was just wondering if there's any sense you have that with the climate change now seeming to be accelerating, we'll see that happening, whether there's any sense that the invertebrates could adapt to actually shift their optimal temperature for reproduction upwards to cope with that.
I think it's hard to tell from our data set, but there is good evidence that the optimal point of that curve, the peak reproductive performance can shift, and we have lab studies showing that. Whether it can shift fast enough for the rate that the climate is changing, I think is unclear. I suspect not. I think that's what the evidence suggests, that it's shifting. I mean, there was a recent meta-analysis, I think, that showed that every degree in exposure temperature, the peak changed by like 0.1 of a degree, right? So it's shifting, but it might be like 1/10 the rate that it should be. That's hard to tell from our data set specifically, but I think the answer is probably not.
Thank you.
Okay, thank you. I see more questions coming up, but I suggest we deal with them later, and we just follow the scheme and give the word first to Amandine. So I see your slides already arrived, Amandine, so that's very nice. So I give the word to you.
Sorry, I'm mute.
Okay, no problem. Go ahead.
So thanks a lot, Hélène, and good morning or good afternoon to everyone. So I'm going to present you as a network we have been organizing since 2017 now, with a bunch of guys you see here. I'm going to explain further that in the next slides. So this network is on the ecological genomics of coevolutionary interaction. And we proposed this network following one workshop we are initially organized in Zurich. And from then we say, okay, that could be nice to discuss this topic for the next year. So I want to thank the ESEB for the funding and also other local fundings. So if you have any question, please do not hesitate to contact me at the end and also by mail. And also we have a website. It's not a .org, but it's a wiki website. So symbiotic interactions are shaping diversity in ecosystem. We know that now. You have host-parasite interaction. You have also host-mutualist interaction. But surprisingly, we still don't know how many genes and which genes are really involved in host-symbiotic interaction. And this is mostly because we are missing data at the genome side, at the genome level in both sides of the interaction. So in the, for instance, parasite side or host side. And what we need to answer this question is to combine theoretical approach and also experimental approach to really understand the evolutionary process shaping coevolution. So based on this observation, we start to discuss all together with so Daniel, who is at University of Neuchâtel, Dieter, who is at University of Bern, Peter from University of Minnesota, Aurélien at Technical University in Munich, and Eva at Max Planck Institute. So we first met in 2017 for a workshop, and then we say, okay, there were a call by the ESEB, and maybe we can form a group to discuss this topic. And actually, so how groups work from theoretical to experimental approach on various model systems, from animal system to plant system, so it can give a broad overview of process of coevolutions. So, so far we have organized 4 workshops on site. The first one was in France, is Gis-sur-Yvette, so just right next to here. Then in Germany at the TUM, and then also in Germany in Berlin at the WICO. Maybe some of you know this institute. It's Institute for Advanced Study. And then we went back in France last year for a young scientist workshop, and I will show you a bit more on the next slide about each of these workshops. And of course, during the lockdown of COVID lockdown, we had some problem to organize on-site workshops, so we did online seminars. And we plan to do a final conference at the ESEB 2025 conference. We are still discussing with the main organizer to see if it's possible and how we can do a satellite meeting. So we will keep you posted about it. And to spread the word of these different events, we use mostly Twitter, and also we have a website, and of course we use our different network. So the first workshop was actually a kickoff workshop, and the aim was to train young one on experimental approach to test for coevolution. It was a 4-day workshop at GIF-sur-Yvette, so we have invited several speakers from UK, Finland, Germany. We wanted to avoid the carbon footprint, so we didn't invite people from a very far place. And we were discussing a lot of different systems, so from animals to plants to microbes. And so here, so you see it was a GIF in the castle of GIF, a typical French site. And so you see Peter, Daniel here. Each of us were presenting what their group were doing, and also we invited a young researcher, and their travel was paid with the ESEB grant. We have also had additional funding from local research group, and also University Paris-Saclay and so on, to complete the ESEB funding. So we presented the network as well, and it was the first time to brainstorm about coevolution, genomics, which tool we could develop or are already available to test for coevolution. So this was a very interesting workshop, and it raised a lot of questions, and we say, okay, let's see us again the next year, but to discuss this time more theoretical approach to test for coevolution. It was a 3-day workshop with in the morning courses, conference, and in the afternoon practical courses. So we had a bunch of young scientists, we were experts in a theoretical approach to test for coevolution, including Aile, Maoyan, Maike, and Hildegard, and also Mike Booth, who is an expert of theoretical modeling of coevolutionary interaction. So after the completion of these 2 workshops, we had brainstormed a lot, and we decided to start writing a review on this topic. It was just like an emerging idea, so it's starting from that day. And yeah, so we decided only between the PI of the network to discuss this topic in Berlin, to further dig in this question of the genomics of coevolution. So Mike and Britt and Jessica were hosted by the WICO. So we took this opportunity to have a funding from the WICO as well to complete the ESEB funding to brainstorm about the idea of the genomic of coevolution. And then last fall, we proposed to the young scientists for each of our groups to organize a meeting, and so we did that at the EDEV, which is our new institute, the Institute of Diversity and Ecology and Evolution of the Living Planet. And I really want to thank them all because they did a very great job to organize this meeting, and there were a lot of brainstorming. It was also a big network event for them to know each other and to develop a community working on coevolution. And as I told you, we did also several online seminars in 2020 and 2021 to brainstorm on this idea, and also to still keep the pace of the network despite this lockdown. And after all this brainstorming and discussion, so we published in 2022 a review in Trends in Genetics. And so basically we propose some idea on which process can shape coevolution, which data are missing. So I'm not going to detail all this figure, but we detail all the major factors underlying coevolutionary dynamics in an ecological context and what was missing so far in the literature, and some practical guide if you want to really study coevolution based on genomics data, but also ecological data. And our final step of this network will be the ESEB conference. I cross my fingers. So we need to confirm that with Tony Gabadon to see if it's possible to do a satellite meeting. And so we will announce the speaker list soon. You will know that on the different social media and also on our website. And on that, I want to thank you a lot for your attention, and also all the organizer and also all the participants to these workshops, and especially the young scientists, because the aim is also to build a community that can continue working on this question. And also Ute, Franco, and Hélène for all their help and their patience. And if you have any question, you can contact me again, and you can also go on the website, and you will follow. You can follow also the update of this network on this Twitter hashtag. So thanks a lot, and I will be happy to answer any question.
Okay, thank you, Amandine. You're very much on time, so that's good. We still have 3 minutes. So I'm looking if there are some questions. Anyone that has a question? Maybe I will ask a first question. So on the general overview, because you went very fast over the paper, maybe you can go back a few slides.
Sure.
Yeah. So what is your main conclusion here? So what are the next steps to take, you think, based on this study?
Yeah, that's a very good question. So the idea is to say, okay, so when people want to test for coevolution, most of the time they only look at one side, and actually you really need to combine data on both sides. But there are several challenges to do that. First, of course, the cost of sequencing, but there are also a limitation in the theoretical concept behind. What do you expect in terms of footprint of coevolution? At the genome level, you have a lot of different models, but at the genomic level, we still don't have a very good idea of those footprints. So there are some approximate Bayesian computation developments that are proposed, and mostly by Aurélien, the Telliers team, actually. But what we're still missing, it's really the data in both sides and some theoretical model behind. And also what we say in conclusion is that it's not only about the genomic data, but it's also the sampling you do. And as coevolution, it's a dynamic process. You need to sample at different time. If you only look at one time in the evolutionary time scale, it's only one shot. So if you are lucky, okay, you fall on the footprint of coevolution. If not, and depending on your model, you won't fall in this footprint. So 2 ways to overpass this problem is either you sample in time, but it's not always possible for all models. Like I'm working on trees, it's very complicated to sample at different time, or you can sample at different in different space. Like if you do a sampling across Europe, you can have different population in different colonization states, so it can represent different time scale. So the conclusion, it's like, okay, more genomic data, but we need more models, and also more sampling, deep sampling, to have an ecological view of coevolution.
Okay, thank you for that clarification. So in the meantime, so Ed, can you load your presentation? And I will look if we have some more questions. There's a question, but I don't see from who it's coming later.
It's from me.
Okay, go ahead.
I just wonder, how is your model, can it be converted to sexual conflict as well, and the co-evolutionary dynamics of sexual conflict? Or does it need 2 species?
No, actually, yes, we say 2 species. So I'm not sure about sexual conflict. Maybe it could be. And to be honest, it's much more, I mean, theoretical model, it's much more Aurélien, who is the expert. I'm much more the biologist on the field. But clearly, we start from 2 species. But what we explain is that, okay, we need to start and from host-parasite interaction, because this is from this system that we know more. But actually, we need to move at the community level. And I mean, people work on microbiome. We discuss a lot what to expect at the microbiome level. But so far, the model look at 2 species interaction. And we really need to move forward to multi-species. But of course, we need to start from the easiest. That always the most problem. But there are more and more models on that, but still.
Okay, cool, thank you.
Thank you.
Thank you very much for that. Okay, so thank you. And then we go over to the next STN, which is on micro and macro evolutionary processes shaping prokaryotic genomes. So I give the floor to you.
Thank you very much. Thanks, everybody. Very interesting talk so far. So I'll just introduce myself. So I'm a professor at the University of Bath in the west of England. So most of my research career has been on looking at pathogenic bacteria, specifically bacteria pathogenic to humans, certainly, but also more recently to for animals, and looking specifically at this increasing problem of antibiotic resistance. So my research has essentially been more public health facing than fundamental evolutionary, but I've always been interested in evolutionary questions since my PhD, and the fact that we've got so many such large genome data sets now, primarily generated for public health reasons, but also provide absolutely amazing resource for addressing more fundamental evolutionary questions. So the history of this this network, goes back to 2017, actually, when I met Deepa Gashi, who's in Bangalore in India, at an SMBE satellite meeting that I arranged in India. And this was really focusing on how we can begin to pull together all the evidence from experimental evolution experiments. I dare say you've all heard of the long-term evolution experiment, the Lenski lines, this experiment that's been running from 1988, where cultures just passage daily, and we can freeze down the cultures and can understand the how repeatable the responses are to those conditions in the lab, and understand very fundamental evolutionary processes. So I was really interested in how experiments like this, and there are many, many other experimental evolution studies, of course, addressing different, begin to reconcile the kinds of inferences and questions that we're getting from those studies with what we're actually seeing in natural populations, the kind of genome data that I was used to staring at. So a couple of years later, we along with Ruth Hirschberg in Haifa in Israel, Isabel Gordo in Portugal, and Olivia Teneon in Paris, all of whom are absolute experts at experimental evolution and evolutionary processes. We decided to, we were awarded this network titled Micro and Macro Evolutionary Processes Shaping Prokaryotic Genomes. Now, it's a very, very broad title, and we gave ourselves quite a lot of slack, I guess, in our initial remit. But this is really just to give you some idea of the amount of data that we had to data from natural populations that we have to work with. So just recently, a colleague in Bath, Zamin Iqbal, has released this resource where basically there are assemblies for almost 2 million genomes readily available. It's called, neatly, it's called all the bacteria. It's currently in the archive, so you can pull out nearly 2 million genomes of different bacterial species. And it's not just the numbers of genomes that are available. The COVID, partly due to COVID, there was a quite rapid development in long-read sequencing technology, particularly this nanopore platform. And what that means for bacterial genomes is that we can actually have properly complete genome sequences. If we combine the short-read data with the long-read data, we can build complete chromosomes, and just as importantly, all the extra chromosomal elements, all the plasmids, which we haven't really been able to work with before in a sort of population scale. Now suddenly we've got data sets for thousands of isolates, fully completed genomes, each one having an average 1.52 plasmids per cell, so thousands and thousands of plasmid sequences. So this opens up a whole new avenue of research and population dynamics. So the overarching aims of this SCN, as expressed in the original application, to understand patterns of variation within and between prokaryotic read bacterial, frankly, populations through synthesis of genomic and population forces, operating over very different timescales. So essentially, to equate this with what I was saying earlier, the short timescales would be the experimental evolution, and the long evolutionary timescales would be from natural sequences. Although, of course, because we've got the ability to generate genome sequences so rapidly, we can also look at very short-term evolutionary changes in nature. So how do we bridge this gap between what we see within populations, the effects of drift, within populations, the population size and so on, with what happens over macroevolution. It's a fundamental evolutionary problem. These two things seem to be kind of difficult to reconcile. So this was where we started from, and of course, this being such a broad remit, we inevitably sort of funneled it down to more sort of detailed and focused questions. So as with other networks, we had to sit on our heels more or less for a couple of years. That we planned the initial meeting for 2020. Obviously, that didn't happen. So our kickoff meeting happened at my host institute, the Milner Centre for Evolution at the University of Bath. It's a 2.5-day workshop from 30th May to 1st June in Bath. We had 33 participants. We spent quite a lot of the budget bringing people over. We wanted it to be geographically diverse. So we had people from not only Europe, but Asia, well, deepest from India. Obviously, we had other Asian colleagues coming, and North and Central, quite a lot of colleagues from the States and from Central America. So as well as the 5 organizers, we had 12 invited speakers. And then our strategy for the all-important component, the postdocs and the postgrads, was that we invited applications for them to send in an abstract to come and give a talk and participate in this meeting. We received 27 good applications. They were all pretty good, from which we whittled down 16. So we paid for those guys to come over and join in this workshop. So these are the very professionally produced name badges from the workshop. So some of those names may be familiar to you. So really high profile list of speakers. Jeff Barrick at the top there is actually now managing the long-term evolution experiment that I mentioned before. So the topics that were covered in the talks from the invited speakers were quite broad, covering all sorts of aspects around evolution of genome size. Antibiotic resistance, of course, always comes up when you're talking about anything to do with bacterial evolution. We had quite a strong focus on plasmids and other mobile genetic elements, and mutation rate and mutation biases, and of course, how to measure selection in both natural and artificial conditions. So on the final day, we split into 2 working groups, and we essentially had 2 different topics to discuss on how we could take this forward. One, we decided to focus specifically on the questions of mobile genetic elements, plasmids, and their role in bacterial adaptation. So clearly they carry genes that are adaptive to bacteria, but they also have, it's one of these questions, well, where is selection really operating? Sometimes it's operating in favor of the plasmid, sometimes it's operating in favor of the host, sometimes both. So disentangling all these selective forces was really at the crux of what we were discussing. And related to that is how these so-called cryptic plasmids persist in bacterial populations. There's a number of interesting models now come to light to try and explain this. So these aren't conferring any obvious advantage to the bacterial host. A very interesting, very complicated feature of plasmids is that they not only undergo single nucleotide polymorphisms very quickly, of course, but they also undergo a massive structural rearrangements at very high speed, and this is probably more important in terms of their, certainly in terms of their short-term evolution than the mutations they pick up. So how to incorporate these, so there's a fundamental question here of how we incorporate these structural rearrangements into evolutionary models to try and see if we can reconstruct any sort of meaningful phylogeny. But also a much more practical reason is the question of when are 2 plasmids the same? How many rearrangements do they have to encounter before we can count them as different plasmids? And this sort of question is important from a public health point of view, because we're interested in plasmid carrying antibiotic resistance genes, maybe spreading through hospitals, for example, independent of the bacterial host. So if we want to understand how they're spread, we have to agree on when 2 plasmids are the same thing in order to infer a single transmission event. So these aren't just sort of blue skies questions. These are really important concepts and challenges for public health when it comes to plasmid epidemiology. So the other working group was looking at the mutation biases, and a lot of us, myself and Deepa and others on the organising committee, have an interest in GC content. There's a basic problem with GC content with bacteria, and that it tends to be too high. If you look at what mutation biases will want to do, is to push GC content down to maybe 20% GC. There's a bias from GC to AT, but nevertheless, we see much higher GC contents than that in most bacteria. So what's maintaining that high GC content? And this is important. This may have been the fundamental question for evolutionary innovation, for gene expression, and so on. So Deepa in particular has done some really interesting work subsequent to this this first meeting on looking at how different bacterial lineages can suddenly jump in GC content, and this can open the door to new sort of avenues of evolutionary innovation. So this is a slightly less optimistic graph, which I came away feeling about after all these discussions, that actually not only are plasmids very hard to classify, very hard to model from an evolutionary sense, they're even quite hard to define. And there are elements that are sort of half plasmids, half phage, and some of my preconceived ideas about biology of plasmids really fell away pretty quickly after this meeting, which is a good thing, of course. So it wasn't all work. Of course, we had a very nice evening on the River Avon, which runs through Bath. We had dinner on a barge. This is Olivier and Tal Dagnan and me in the back there, and Jeff Barrett. The poster boy from this initial meeting was James Horton, who's a postdoc in the at Bath, who came to the meeting, made a big contribution to discussions, and he actually went away and decided that he wanted to combine these two themes of mutation biases and mobile genetic elements, and look specifically at where the mutation biases of plasmids actually mirror the mutation biases of the host, which is an interesting question because it could tell us something about the host range and the history of specific plasmids in terms of how they've moved around between different hosts over evolutionary time. So he went and designed a really elegant experiment to test this, which is now being written up. It worked pretty well. And he's now got awarded an EMBO fellowship, partly on the back of that, and he's now working with Ivan Matic. So that was a really nice direct result of that initial meeting. We've also got an opinion article, which is just got reviews back this morning, which are quite favourable, from Microbial Genomics, which is relating to this question about how to quantify the evolutionary distances between different plasmids. So going forward, our next meeting will be in Split at the Mediterranean Institute for Life Sciences. This will be held very likely in the last week of October. We're going to have more or less the same model as we did before, so we will be putting out a call for abstracts to be submitted from early career researchers, from which we will select the best ones, as many as we can afford. We'll fly over. This hopefully this meeting will have a greater emphasis on the training component, and will be more focused on following up from previous discussions, looking specifically at plasmids and mutation biases. And that's okay. Thank you very much.
Okay, thank you, and thank you for sharing all these ideas. So thank you very much. Yeah, are there any questions? Maybe in the meantime, Franjo, you can already also load the slides of the next, yeah, loading okay. No questions so far, but you can put them in the chat or in the Q&A, and then we can still discuss further later. So yeah, then we go on with the Wild Animal Microbiome and Evolution Special Topic Network, which will be presented by Franjo, who is replacing Sarah. You'll find you're still you're still muted, Franjo.
Yes.
Yeah. Okay.
And now I couldn't find the unmute button. Can you hear me now?
Yeah, we hear you very well.
Okay.
Yeah.
So I'm not Sarah Worsley, who was supposed to present the wild animal microbiome and evolution special topic network here. But this morning, she informed us that she has caught COVID and that she is not feeling well and therefore cannot give the talk. But she sent me her slides, and I have very limited knowledge about microbiomes and so on, but I know a little bit about their network, and I will try to walk you through the slides and give you an impression of what this network has been doing and why it may be useful for you to participate as well. So the name is WAME of the network, and it is about wild animal microbiomes. I think nowadays it's not necessary to say that the microbiome, be it the gut microbiome or the skin microbiome or the oral microbiome and so on, that they can all have very important effects on the host, on cognitive functions, about digestion, health, and disease, and all kinds of things. And therefore, it's also not surprising so that the literature on this topic is exploding. So it shows an exponential growth, the number of papers in this field. However, most of the work that is being done is on either on human microbiomes or on lab animals like these inbred lines of house mice. And this is a certain problem because these lab animals, they are housed under very specific conditions. So in sterile environments, and they are typically then living under highly constant kind of conditions. They often are also kept healthy by antibiotic treatments and so on. And as I said, that they are typically highly inbred and therefore have a very low diversity. And there's quite a number of studies nowadays who show that it is problematic to extrapolate from these lab animals to animals in the wild. And that's basically the thing that WAME is then looking at, namely the microbiomes of wild animals. And one of the things that what has already been remarked earlier is that the characteristics of the microbiome are highly species and also clade specific. So here is, for example, a graph that's showing that for very different kind of organisms, the signature of the microbiome, which is of course not trivial to determine, because the microbiome is of course consists of many kind of components. But if you try to classify the signature, then you see quite some diversity across the animal kingdom. And that's one of the questions that people are asking then in our field from the eco-evolutionary perspective: what drives this microbiome variation? But also, if you look into a little bit more within species kind of comparisons, then you also encounter quite some interesting questions. Here, for example, data on baboons, where then you have some longitudinal studies of microbiome similarity. And the upper curve shows then the similarity of different samples from the same individual over time. And then the green one shows then the similarity of an individual with a group member, so a different group member, and the signature of this microbiome of the group member over time. And then the red line shows then something like an extra group member. And you see that there's a clear relationship there, but it's also not very clear what exactly this represents. And for us, from an evolutionary perspective, the questions are also, what are the consequences for evolution and also for ecology, for host fitness, and things like that. Now, if you now want to study microbiomes in the wild, then you encounter many problems in addition to the problems that you already encounter with lab animals. So I already mentioned it's not trivial at all to quantify or to say something what the signature of the microbiome is. in particular if you want to do it across species. But then, of course, also a lot of logistic kind of problems that I cannot judge at all, but I can imagine that the logistics of sample collection in the wild can be quite problematic. And also the sample storage can be problematic. And then you need to extract then DNA, often then under field conditions and things like that. And then choosing the right kind of omics technology, and also to compare then different kinds of studies with each other is all relatively challenging. And of course, fortunately, the approaches are then very rapidly evolving, but it's also relatively complex. And it's very difficult for a field researcher to keep well informed about what's happening in the field. And that was actually one of the reasons why then WAIM was established then in 2021. Basically, the idea was to bring together very different researchers who could then exchange their expertise and their knowledge with each other, discuss problems, methodological problems, but also scientific research questions with each other. And the other thing was that one was feeling the need that it is really important for, in particular also for field workers, to provide a support network, particularly for early career researchers, because these microbiome techniques are not trivial to use, and need a lot of know-how that you often do not have if you are somebody working then in the field with animals. And of course, in the end, the idea was also then to shift the whole field forward and to be able to say a little bit more about the role of the microbiome for the wild animal ecology and evolution. Now, WAIM has an organizational committee. In this case, there are 12 different researchers. The text here is much too small for you to read it, but you can look them up on the website. The most important point is that they have collected a group of people with very diverse expertise. In particular, they're working on very different animals. So some are specialists on mammals, others on birds, others on fish, amphibians, invertebrates, you name it. So this also allows them to be able to host quite a number of early career researchers and also to exchange their insights and ideas with each other. Now, they have been quite active in the meantime. So they've organized, for example, several seminar series. So there was one about host specificity of the animal gut microbiome. Then there was one on longitudinal dynamics of the microbiome and microbiome mediated behavior, and also conservation issues related to the microbiome. And they also organized a symposium at the last ESEB conference in 2022 in Prague. And so they also have published together. So most important thing is perhaps a special issue of the Journal of Evolutionary Biology, which is then about evolutionary patterns and processes in animal microbiomes. But they also have jointly written several papers together, and one of them is here shown as a representative example, which is now on EcoEvo RXIV in the preprint server. Regarding the support for early career researchers, they really try to provide them the opportunity to learn the most important techniques. So they, on the one hand, they have a Slack channel, so where protocols are being published, analytical methods are being published, where the important literature can be found. Also, career opportunities are posted and so on. And they also organize some meetings. But very important and very interesting is perhaps also the possibility that the network funds then some lab visits for early career researchers. So they can sponsor a trip and a lab visit to one of the WAIM groups. So they for this they have an application round. So they will at this moment, for example, the 2024 round is currently being assessed, but they also expect to fund a future round in 2025. So if you are in the if you belong to the target group and are interested in a lab visit to one of the groups associated with WAIM, you should contact one of the organizers. In addition, they have, of course, some future plans. So they want to continue with online seminar series. And if you have an idea about an interesting seminar series about a topic or something like this, then, or if you want to organize one, you should get in touch with the organizers. And then last but not least, so they also plan to organize a symposium and a methods workshop at the next ESEB Congress in Barcelona 2025. Now that was it more or less. Further information on WAIM can be found on the website. They also have then an X account, a special Gmail account, or you can also contact anybody of the organizing committee. So unfortunately, I couldn't tell you anything scientifically and any insights and so on. So I'm just not sufficiently into the subject to be able to talk about these things. But at least I think you've got an idea of what this network is doing. Thanks a lot.
Thank you very much, Franjo. You did a terrific job, especially because you received the slides like 2 hours ago or something. So that's really great. It's also great to see all these activities. So it's really nice to see that all the STNs are so active and that there's so much networking, especially also with chances for early career researchers. So that's very nice to see. So yeah, I think we can go over to the question and answer session. So so if people feel like asking a question, so don't hesitate to put it in the chat. So maybe we can go already over a few questions that are there. I think for the fertility, thermal fertility network, there were some questions, if not mistaken, but some have been answered already in the chat too. But so Rhonda, you had a comment, right? Rhonda, you're still there? I think you are. Yeah.
I am, but I don't have a comment.
I saw something on the protocols that you wrote.
Oh, yes. Yeah. I just wanted to say that in addition to funding just a general protocol for small invertebrates, other members of the STN generated modified protocols for their organism of choice, most of which were vertebrates that have regulations about what you can and can't do with them. So in addition to the 3 protocols, there's also modifications. So take a look at those appendices if you're interested.
Thank you. And then there's a question from Amanda. Amanda, are you still there? Yeah, I see you. Yeah.
Yeah, it wasn't a question. I was answering. So there was some talk about how evolvable reproduction is compared to lifespan, and that's sort of linked to Jen's question about which we would expect to be, or how the declines would work together. And in terms of the limits, what we know so far from Van Heerwaarden and Segro's work is that those temperature limits don't seem to be very evolvable. So that's probably bad news. But I would say, again, this was a Drosophila study. And yeah, finding out about more animals would be really helpful.
Yeah, thank you for that. Yeah, and indeed, I skipped Jen's answer. So Jen, I don't know if you're still there, but if you want to add something to that or not, or if someone from the STN wants to reply on that.
Yeah, the question was just about could survival can't be more sensitive than reproduction, right? Because you have to be alive to reproduce. So we're only looking in one direction. So I think that was the question, and my answer to that is yes. The question is, how much different is reproduction? from survival? How much stronger is the sensitivity? I think that's the key question, because people often don't measure reproduction at all. So if that disparity is big, then we need to know about it because we need to start measuring it.
Okay, thank you. Daniel, you want to say something?
Yeah. Yeah, I also have a question to your thermal limit STN. So you started first with the limits, so the limits of fertility, but then you changed the more or less the focus more on life history, so on how then survival and reproduction profiles change with temperature. And I think this was a wise choice. But my question is, to what extent can you then still say something about then the limits of say of species abundance in the context of global warming or things like that? So to what do you then need something like a limit approach, or do your profiles also tell a whole story?
Maybe I'll jump in first. I think we need both. I think it's sometimes hard to tell. We need to start to recapitulate the performance curve to even predict where the limit's going to be, right? So often the performance curves have a similar shape, but we need to get at least high enough that we can start seeing a considerable drop. But I also don't even think. I think we really do need limits as well. I don't think we can tell everything just from the shape of the thermal performance curve.
I think that the choice of the performance curves is really a good one, but I could imagine that it very much depends, for example, on the productivity of a species. So if you have an insect species, of course you have mainly insect or invertebrate data at this moment, but that the same kind of curves might mean, ecologically speaking, something very different for species with quite some high abundance or lower abundance.
Yeah, sure. Linking that to fitness changes is going to be difficult at the minute, I think, and we need to measure these things outside in the real world, I think, as well. I agree.
Okay, and then a comment from Ophélie, a question from Ophélie.
Yeah, I had a question actually to all participants to those STN. So I must say it's really great to see all this activity, and I think it's been super helpful and productive for the community to have those networks, and this is really exciting. And I would like to remind that the sponsorship of those networks come from ESEB money, and ESEB money comes from the journal JEB. I mean, all the income of the society comes from the journal. And given also, debates concerning publication and where our publication money is going and whether it's feeding back in the community, I can say that those STN are a way to feed back the money from publication into the community in a very efficient and fruitful way. And so I wanted to know how in those different networks you discuss where you publish your work in this context, and what kind of decision and trade-off you make with regard of publishing maybe in the most adequate outlet for your specific research, and publishing in a journal where the money for publication is going to be reinvested in the community you benefit from. Anybody wants to answer?
That wants to take that. So I saw the Wayne had a special session, a special series based on, I think also the symposium they had in Prague. So based on that, there was a special session. So that's definitely an opportunity to consider if people want to do that. Any other?
Oh, if I may.
Yeah, sure.
So I think we're all grateful to ESEB, and so for example, our first paper, well, the second was published in the Journal of Evolutionary Biology, and also to acknowledge the support that we received. And I guess the other one that we might consider for the future is evolution letters. But also we have a feeling it needs to fit within that. So the map, for example, we didn't felt was a very good fit, so we went for a different journal there. But we definitely considered those journals and have actively used them.
Cool. Any other comment, reaction, or question? Franjo?
I've posed a question to this to the ecological genomics of co-evolutionary interactions no, was Amandine. Amandine, the point. So you mentioned how difficult it is to really answer the question to what extent then at genomic level you have really something like a fit between two different species, and also to infer something like causality and so on. I wonder whether there are in the meantime 2 or 3 example cases, so where the genomic kind of processes have been unraveled in quite some detail. I am a theoretician, and I'm typically using very simplistic kind of models with the gene-for-gene interactions in 2 different species and things like that, which are probably completely wrong. But where can I find information that tells me that I'm wrong in my old-fashioned thinking? And is something like this already available at this moment?
Yeah, it's not old-fashioned. It's just that there are some typical case of gen-for-gen interaction. You have the Daphnia model, for instance, for which has been very well documented. It doesn't mean that there is no any study. It means that you have very, very few case studies that really are well documented. So the Daphnia model clearly is the one which is the best documented. I can send you several examples by mail as well. And actually, we document them in the review, which is not in Journal of Evolutionary Biology. I wanted to jump on the question. But I can send you actually some other cases. It's not that it's the literature has not, how to say, you don't have clear case as well because the genomic tools were not available as well. But you have like cases for which it was possible to get the genome because they were small, and clearly the Daphnia system is very nice. But the most tricky part is when it comes to plants and hosts and parasite, for instance, because the plant genome are much bigger. So, but there are some examples. Clearly, there is also a study which is on mutualism. So it's not like full genome of the plants, but at least they show some genes that are involved in the mutualism, which is pretty cool. Mm-hmm. I'm very focused on mutualism, so that's why I love it. But I can send it to you by mail, and we also put it in the review. Yeah. And so sorry, Franjo. And why we did choose transient genetic, because it was a very genomics discussion. And it's also about which audience you target. And at the time when we had this ESEB consortium, which has been built, this question of feeding back the Journal of Evolutionary Biology, the ESEB, was not really clear to us, to be honest. And actually all these PCI frameworks that I'm really in, and I'm publishing in those journal right now, we were not really in that part, and we shaped the review initially for the TISH paper. So, I mean, there were a lot of questions on that part. Do we change everything? But we are planning also to publish this in JIB. So it's on the way, I would say.
Cool, nice to hear.
Thank you.
Anyone else that wants to react to that, or has another question on of the other networks? No, if not, you can still. No, we go to another session. The session is in another, so you won't see it in the chat. Anyhow, if you would still have a question, you can always come to the next session, who is starting at 5 o'clock in European time and then 11 o'clock in American time. So very welcome to that. And then we will have another overview of different networks. So thank you very much all for participating also in this discussion. And thank you very much for all the work. It's highly appreciated.
Thanks a lot, Tom.
Yeah.
Thank you.
See you in half an hour.
Bye.
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 Thank you.
Yeah.
Bye. Thank you so much.